Hello everyone, good afternoon and welcome to today's live broadcast of the third part of the three-part webinar series on digital RNA sequencing. I am Krishnan Alampalam and I am your moderator for today's event. Before we begin, we have a couple of announcements. All the products described here are for research use only. They should not be used for diagnosis, treatment, or prevention of any disease. For any additional information, please visit us at www.QIAGEN.com or talk to your technical support team. To ensure that everybody can hear the speaker clearly, we have muted all the inbound lines. So for any interaction with us or the speaker, please use the questions box that appears on the lower right-hand side of your monitor. If you have any questions or any technical problems, use this questions box. Just to make sure that you can hear me clearly, please enter yes or no in the questions box. Okay, thank you. This webinar is designed to be interactive, so please feel free to ask questions anytime during the webinar. Do not wait till the end. With that, now today QIAGEN is very excited to announce the launch of QIAseq targeted RNA panels. These have been developed as a sample to insight solutions for quantitative gene expression profiling using digital RNA-seq. There are many questions about this technology. What is digital RNA sequencing? How does this differ from standard RNA sequencing? Why one should use digital RNA sequencing for gene expression profiling? How to plan a digital RNA-seq experiment? How to analyze the data? To answer all these questions, we had this three-part educational webinar series, of which the first part was given on February 17th by Dr. Eric Lader, which is what is digital RNA-seq. The second one was given by Dr. Raid Samara on digital RNA sequencing for gene expression profiling. And the third one today is being given by Dr. Melanie Husong, scientist NGS, QIAGEN, and Dr. Jean-Noel Billard, principal scientist in the bioinformatics team. With that, let me introduce to both the speakers today. First is Dr. Melanie Husong. She will start this webinar series. Dr. Husong is an experienced molecular biologist and a genetist with 12+ years of experience. She is very knowledgeable in the area of next-generation sequencing and its application in clinical diagnostics and research environment. She received her PhD from University of Melbourne, Australia, and has published numerous papers and presented extensively in this field. Prior to joining QIAGEN, the results of her research and development products were groundbreaking and led to prominent diagnostic tests using a variety of NGS library prep methods. Dr. Husong is a recent addition to the QIAGEN family and is currently working on developing new products for RNA sequencing. The second speaker for today is Dr. Jean-Noel Billard, principal scientist at QIAGEN Bioinformatics. He joined the Ingenuity Systems, now a part of QIAGEN, in 2008 as a staff scientist for in silico research program in oncology and infectious diseases. Jean-Noel holds a PhD in blood cell biology from Paris 7 and has done his postdoctoral work at the Scripps Research Institute. With that, let me introduce you to today's title, Molecular Insights into Gene Expression Profiling Using Digital RNA-seq Data Analysis Tutorial by Dr. Melanie Husong and Dr. Jean-Noel Billard. Take it over from here, Melanie.
Thank you for the introduction, Krishnan. And so as Krishnan just stated, we're going to be going through a data analysis tutorial today with using the molecular insight into gene expression for digital RNA-seq. So on today's agenda is why are we using targeted next generation sequencing to study gene expression? We're going to go through targeted enrichment using the QIAseq targeted RNA panels. We're going to discuss the QIAseq primary data analysis and go through read mapping and the molecular barcode counting. And then we're going to go over the QIAseq expression analysis and discuss the workflow and the interface for doing the fold change calculations for RNA-seq. And then there's Jean-Noel will be talking about the data interpretation that can be done in the Ingenuity Pathway Analysis, IPA software. So gene expression profiling is central to many biological processes. That is the reason why we want to study gene expression. So I'm a scientist and I have 96 samples and I'd like to investigate 500 genes, but I only have a limited amount of total RNA, say 25 to 50 nanograms of each of the samples. And my research budget is really low and I can only afford about $100 a sample. So what do? Do I follow up on other experiments that I have done? What gene expression profiling data or methodologies can I use? There are PCR-based methodologies out there that are accurate, but there's limited sample and assay throughput, and it requires a lot of RNA. Then there's whole transcriptome sequencing, which is expensive, and you get high throughput power, and the data is very complex, and you need a bioinformatician to be able to go through it and help you with the data analysis. Microarrays, they have easy data analysis processes have been made, but they have high background noise, and it requires a lot of DNA and is limited dynamic range. Then there's other traditional targeted RNA sequencing methods where your data is in a manageable amount. It's a low cost per sample, but you have issues with amplification bias. So QIAseq targeted RNA high throughput digital next generation sequencing is an advantage over all of the other methodologies. It's simple to use and all the complexity is behind the scenes. Users do not have to worry about it. It's a complete integrated system from sample to insight. It's sensitive and highly specific. It is extremely flexible in the experiment design. You can do as many samples as you like and study as many genes as you like. It all depends on the research question that you would like to answer. It's simple for the end user to address the bioinformatically because you don't really need the bioinformatician that's down the hall to come and have a look and do your data analysis for you. We've developed a easy-to-use complementary platform. This method requires no ribosomal RNA depletion or blocking or DT selection, and it makes use of limited next-generation sequencing read budget. And you know what sequencing instrument you have, so you know exactly the read depth that you can get for your sequencing. QIAGEN has leveraged the content know-how, and it allows you to have flexible content of the panels, because QIAGEN has been developing panels for years that are disease and pathway focused. So we've developed a test that is ready to use, easy to modify and customize, and it is all content in one kit. So when, who, and why would scientists want to use it? But you guys know what you're interested in. If you're studying cancer or differentiation, you know and already have a list of genes and pathways involved in your mind that you would like to investigate. So you can either follow up on broader experiments, such as the whole transcriptome sequencing, or from microarray. Sorry, computer froze, and now I'm just finding my slide. Not my slide. All right, sorry about that, guys. So the QIAseq targeted RNA workflow is from sample to insight. You have your sample, so you have your extraction from your RNA, whether it be tissue, FFPE samples. You synthesize the cDNA, go through a library construction, which I'll go through in further detail on the next slide. You then quantitate and QC your library, and then you can put it on any sequencing platform. This test is platform agnostic. You can go on an Illumina platform or an ion torrent platform. And then we have complementary data analysis, the primary analysis, which is the molecular TAD counts, and then a secondary analysis for normalization. And then you can take this secondary data analysis and import it into a data interpretation Ingenuity pathway to get insight into the samples that you're been looking at. So how is a QIAseq targeted RNA library constructed? You have your RNA sample. You then go through first strand cDNA synthesis, and then you have a primer extension and molecular barcoding reaction, where you have here you have 12 base pair unique barcodes that are attached to the gene-specific primer. You go through a QIAB cleanup and then do a first stage PCR reaction with the gene-specific reverse primer. And as you can see in this whole process, the gene-specific primers are never in the same reaction together. So they minimize the amount of primer time you get. You then go through another QIAseq feed cleanup and then do a second stage PCR library construction and sample indexing. So here you add the adapters and the indexes to the ends of the library. This way you can index and multiplex a lot of samples in a sequencing run downstream. And then you do another cleanup. Then with the final library prep, you can quantitate it either using qPCR, a bioanalyzer, or a qubit, whatever you're used to doing, and then it's ready for sequencing. But this whole process from RNA sample down to the cleanup prior to library quantification can be done in one day. It can be done in 6 hours. And all of the reagents are supplied for the cDNA synthesis down to the before the library quantification step, all in one kit. So it's an easy to use kit and process for new users of NGS. I'm sorry, my slides aren't forwarding on. There we go. So what QIAseq targeted RNA panels do we have available that have been wet lab tested? We have a cancer transcriptome panel, which is at size 411 genes. We have inflammation and immunity transcriptome with 491 genes. We have signal transduction pathway finder, which is 421 genes, stem cell and differentiation markers with 309 genes, molecular toxicology and transcriptome of 386 genes, angiogenesis and endothelial cell biology with 356 genes, apoptosis and cell death with 280 genes, and then an extracellular matrix and cell adhesion molecules with 437 genes. All of these panels have included the genomic DNA controls and housekeeping genes, which I will discuss in a little bit further detail. But if you look at one of these panels and you're interested in one of these panels, but you find that your favorite genes aren't included in these panels, you are free to customize and extend the panels with an extra 25 genes. And if you're not interested in researching any of these human panels at all, you can customize the panel by selecting from over 500 pathways. So you can really customize the content to address your research questions that you'd like to answer. And this was discussed in last week's talk on how to go and make and design a custom panel. So once you've done your sequencing, you have your files, and so then you can go into this complementary QIAseq targeted RNA data primary analysis web portal, and you can upload your sequencing files, whether they be FastQ or FastQ-GC files from the Illumina platforms or the BAM files from an Ion Torrent platform. You can manage these files, you can delete them in the File Management tab, and then you have the RNA Quantification Jobs tab, and this is where you submit your runs for analysis and view and download the output files. So in the RNA Quantification tab, you must select the appropriate catalog or custom catalog number up here, and then you go through and you select the samples that you're interested in analyzing. And this is one of the most important steps as well, is that you have to select what lane to use. You have to select lane 1 for MySeq, QIAseq, and Ion Torrent users, and for NextSeq, you have to choose 4 lanes. And the reason why you have to choose 4 lanes for the NextSeq is that although the NextSeq only has a single lane, when you actually download your FastQ files, it has that each of the individual samples has 4 lanes. So the actual analysis will go through and concatenate all of the data, so that when you look at the data afterwards, you don't have 4 columns worth of data for one sample. It combines all of the data together. So that's why it's important to pick 4 lanes for the NextSeq run. Then you create the job. And while the job is running in the background, you go through adaptive trimming of the FastQ files, And then we have read mapping, where we align to this reference sequence that we're interested in. And then also we count the molecular barcodes, and that gets merged with the mapped reads. So reads that are less than 55 base pairs in length after adaptive trimming are likely oligodimers, so they get dropped. The first 12 bases are removed from each of the reads because these are the molecular barcode sequences, but they do get appended to the read identifier. And then STAR RNA read mapper is used to map to the reference genome. STAR uses the gene code transcript models to aid in aligning reads that cross introns. And if reads don't map very well, then they get dropped. So we like to, when we process the read alignments, we like to have at least 30 base pairs of the endogenous RNA sequence to be present in the read. Otherwise, it gets dropped. We like to also drop the off-target or unintended primed reads. And then if the genome alignment at the primer start site is not very good, then these reads are dropped as well, because the boundary between the MT region and the primer region is ambiguous. And this helps us improve the MT counting accuracy. So if we have this transcript here, we've got 5 transcripts, but these bars at the end here represent 1 molecular tag. This lot of transcripts only gets counted as 1 MT. And the reason for that being is that it suggests that with 1 molecular tag that they all originate from 1 molecule. Whereas down here, if you've got the same transcript but with different molecular tags at the end, then you have an MT count of 5, because all of these tags are unique, and that suggests that it's bound to 1 unique molecule for this gene. So the MT count gives you 5. So once all of this is done, and why this is being done, you can check on the job status here on the RNA quantification jobs tab and see where you are in this process. And so hopefully your analysis, you get a successfully done flag, and then you can download the report. And what does this report entail? So this report entails, on the summary tab, the total number of reads that you see per sample, and then you can see that the reads that are dropped because of the primer dimers. If this is really, really high, then it suggests that there's an issue with your cleanup procedure, and you might want to try and go back and improve that. Then you have the reads used per MT counting, and then you have the reads per mean, per MT mean. So what we'd like to see here is that the unique capture reads are roughly between 2 and 3. If this seems to get up to about 10, it seems to plateau off, and you get diminished returns from your sequencing. On the next page, we have your gene ID, your gene symbol, and then you have what gene strand it is on. It tells you the chromosome location and the coordinates for that gene. You have a column that tells you whether it's what the control type is, if it's anything, and then single exon. The majority of the amplicons have a 1, and what this means is that the amplicon that's been designed for this gene is just within an exon, whereas if it's a 0, then that means it's across and spanning an intron. So, and then you have your samples across here, and then these are all the unique molecular tags that are counted for each of the samples and each of the genes that are listed here. So you can actually do a quick look and see that, do an intra-sample comparison of differential genes expression, and then also you can do a quick inter-comparison across different samples of the same gene, and you can see that there's different numbers of molecular tag count. So we also have included in each of the panels the genomic DNA controls. These have been amplicons that are spread out throughout the genome and are in transcriptionally dead regions. And typically the molecular tag count here is very low. If this molecular tag count is high, it gets flagged further down the analysis pipeline, and there could be issues with genomic DNA contamination. Then we also have included in the panels the reference genes that can be used for normalization downstream. So the inclusion of the housekeeping reference genes in the QIAseq RNA panels means that you can normalize the data, and you can also make sample-to-sample and run-to-run comparisons conceivable, because this should hopefully be consistent between runs. So once you've got the data from the primary analysis and the unique MT counts, you can then take this data and upload it into the secondary data analysis setup. So you take the original data and you upload it into this type of format in a, unfortunately, just an XLS format where you've got the gene symbol and then the sample names across here, and these are all the molecular tag counts. And you can put what type of group they go in. So you save that file, and then you can browse here and go and locate the file on your computer, and then press the upload button. And then you get, in the analysis setup tab under the upload data, you get this is how the data was uploaded into the site. So you can double check to make sure that this looks fine. Then you can move across to the sample manager, and in the sample manager workflow, here you can change things if it didn't quite get uploaded correctly. You can change the control groups to group 1, you can change this to group 1, group 2, group 3. Whatever changes you make here, you have to remember to update the changes so that in the analysis they will be recognized. If you forget to do this, then none of the changes will be taken into effect in the analysis downstream. So then you go across into the data QC, and this is for the genomic DNA controls. It will flag and let you know whether your molecular tag count is good or not. So if your molecular tag count is less than 10, you'll get a GC result as good. If you have a molecular tag count that is higher than 10, you'll get a caution down here. And that will just mean that any fold changes as a result may not be accurate with that data that you decide to use. So from there, you can then go across to the select normalization method. But before I go into that, I'll discuss the different normalization methods you can use using QIAseq targeted RNA. So you can use normalize against the unique barcodes themselves if you like, and this method just simply uses the sum of the molecular tags to count in each of the samples for a normalization factor. But this methodology assumes that there are equal amounts of RNA we use in the analysis, and that the majority of the genes are expressed at a relatively stable level across all of the samples. Or you can do the housekeeping genes. You can use one, some, or all housekeeping genes, and these are ranked most to least stably expressed, and the stability ranking is based on a gene norm calculation. Genes that are the stability factor of less than 1.5 are considered to be stably expressed. Or you can use genes of your own choice. And then once you've selected what methodology you want to use to normalize against, the analysis pipeline will calculate the fold change, p-values, generate heat maps, and volcano plots. Of course, p-values and volcano plots can only be generated if you use replicates in your samples. So if I go across into the select normalization method, and in the pull down method here, pick total molecular tags, you can see these are my samples. These are the stably expressed genes and the percent stably expressed, and all of these checked out okay. So I can then press perform normalization, and the normalization process will actually use the total molecular tags. But if I wanted to use the average reference genes instead, I could get this gene list down here with the gene symbols and a stability factor listing. So they're ranked from most stable to the least stable. And here you can select which genes you're interested in using as a normalization factor. If you want to use all 10, then that's okay. Go ahead and select perform normalization. If you want to use just the top 3 ranked genes, you can unselect the bottom 6 and press perform normalization. If you have a custom panel and you decide not to use our control genes, then the genes will end up down here, and you can work out what you would like to use as a housekeeping gene. If you use all of the housekeeping genes, you'll get a fold change that may slightly underestimate the value if you compare that to just using the 3 top ranked housekeeping genes. So once you've decided what you want to do, push the perform normalization step and then go across to the analysis tab. Here you get a list of the genes, the fold change for this is group 1 compared to control, a p-value if replicates were done, and a fold up or down regulation. You can then have a look at this by looking at the plots. You can look at a scatter plot, a volcano plot, or a clustergram, and I'll have an example of those in the next slides. So here's an example of a scatter plot. here are genes that are upregulated in yellow, and down here in blue below the line are genes that are downregulated, and then genes that are unchanged or stably expressed are in the black along the threshold here. You can change the color scheme. You can update what you want as the X and the Y axis, but you've got to remember to press update, and you can export all of this data. And then you have a volcano plot, and you can pick a p-value here, threshold that you want to look at. So these genes are upregulated and are statistically significant because of a p-value is greater than 0.05, and these ones are downregulated. And then clustergram, if you have replicates, you'll be able to see how tight your replicates are, and because hopefully they will come together in the clustergram. You can quickly look and see which genes are more interesting than others. If you have, like these are all your genes, and then you've got 96 gene samples down here that you're looking at, you can pick out which genes are, or samples that might be more interested into looking at. So then you can go across to the export data, and here you get a table of, you can just export the gene table, the IPA upload, fold regulation and p-values, or fold change in p-values by checking or unchecking these boxes, and then you push export. When you set up a secondary analysis, there is no way any of this data is saved with what you've just been analyzing. You have to export the data to be able to get what you've just analyzed. So you get it in an Excel table, and you have different tabs down the bottom. This gene tab shows you what the gene symbol is and then what the catalog number is. You get the fold change in p-value. You get what is being compared at the top, and it says group 1. Your gene symbol, fold changes, and p-values. The next tab along, it's the same thing, but instead of fold change, you get fold regulation. And then the last tab, which is at the front of the Excel sheet, is the IPA upload. And the reason why this is at the front of all the tabs is that you can take this saved export and upload it directly into IPA and interpret your biological findings directly, rather than having to copy and paste things into a different format. And so with that being said, I am now going to handball it off to Jean-Noel, who will discuss IPA.
Thank you, Melanie. I hope everybody is hearing me well. So my name is Jean-Noel, and I'm very glad to work with Melanie on that story about what do we do when we have a small set of RNA that we have sequenced and extracted, and we try to get to the bottom of the biological interpretation. And this is what that slide is representing. So my goal today is in no way try to explain to you what is the breast cancer, which is the example we are going to use, but more so than trying to establish a cool understanding of the fact that QIAGEN has developed that idea of the sample to insight. And what it means is basically from the extraction of your RNA or DNA, this is RNA today, we're able to bring you all the way to the biological interpretations, and that is represented in that slide. But looking at the sample isolation using mini-RNA is the kit that QIAGEN has developed, the QIAseq targeted RNA panel that Melanie has talked about. Then you can enrich with the library construction. You can sequence that by any type of platform you would like. Then you are going to proceed to the data analysis, which Melanie has presented today. And finally, we are going to push that data into IPA for the biological interpretations. I insist on the fact that we are using bioinformatic tools, but I'm in no way a bioinformatician. So what I like about it is trying to be and to the data in a way that I can interpret my biology without having any type of other concern. How we could establish that knowledge? First of all, QIAGEN has developed over the years a gigantic database called the knowledge base. And it's basically coming from data that has been curated from published biomedical papers and peer-reviewed papers. And that findings have been structured over the years and in a way that the software are able to fetch the corresponding scientific findings to highlight that to the scientist in order for him to understand what he has in his data set and be able to bring hypotheses. So what is IPA exactly? Stands for Ingenuity Pathway Analysis. It's a software, it's a solution we have that will allow you to understand many concepts in biology. First of all is the gene and the chemical and the disease view. It's basically when you type a name of a gene and you would like to know about some gene that you have in your data set, we're going to show you everything that we know about that. What the gene regulates, how is it regulated by microRNA, what type of signaling cascade, what we call canonical pathways belongs to, the synonym, and many, many other findings. This is the very strong point of IPA, is able to give you anything you want to know about a particular disease or a disease. We're able to also understand everything about the splicing variant, what we used to call isoform. We have two different models, the RNAseq and ensemble level, and here it's display for caspase 8 all the different splicing variants we have. And when we have findings related to that, these blue hyperlink, you can click on them, and you are going to have the corresponding finding in PubMed. And if you are lucky enough, you get the paper and you get the findings that we have extracted from that. We're able to understand when you overlay your data, your expression pattern of the data set you have on signaling cascade that have been pre-established, what we call canonical, because it's a consensus of a particular cascade. We're able to overlay the data, and we are able even to predict based on the pattern of expression here in green, which is downregulated gene in your sample versus your control, and here it's red, which means upregulated. We are able to predict the general activity of that pathways down to the molecular function here. We're able to strongly also understand the transcriptional program. And I think it's a key message for biologists. And when you have your 23,000 gene, here it's the case of 300 gene in that QIAseq RNA panel, you would like to understand who is driving that, who is the main conductor. And we're able to establish that by reasoning on the genes that are known to be downstream of the particular regulators and their pattern of expression. And we are able to calculate what we call a z-score, and we'll not go in detail, but basically it tells you, based on the value, absolute value, if it's predicted to be activated or inhibited. So it's a key phenomenon that you are going to understand using that upstream analysis. We're able also to understand, based on pattern, again, of expression of your gene in your data set, what biological processes are involved and in what way. Are they upregulated? Are they downregulated? Just to show you, infectious diseases seems to be downregulated. It's a blue color corresponding to a negative z-score, and the cellular movement seems to be upregulated. It's orange, and the z-score corresponds to a value superior at 2. We are able to tie the things together, and I think this is a beautiful image, represent what a biologist wants to understand. If you have your genes in the middle here with their real expression pattern, this is coming from the sequencing. This is what you have from the QIAseq RNA panel here. We're able to understand upstream what are the drivers of that and the prediction of activity, and we're able to understand what is downstream, which we call the downstream effect analysis. We're able to say here that the function of heart seems to be increased based on the pattern of expression of these genes and the upstream regulators. We're able also to understand a relationship between microRNA mRNA. We are not going to develop that here. This is not a topic today, but this is a powerful tool, especially in oncology. We're able to give you also everything you would like to know about a gene and a relation with a disease. So here you type cholestasis, and I would like to say, show me all the gene, all the molecules that have been described in the literature that have been associated to that particular disease. We have the same. We have the isoprofiler. This is not adapted for the QIAseq RNA panel today, but maybe in the future. It's basically telling you and display anything about the splicing variant information you have in your data set and the relative pattern of regulation when you tie that to a particular disease. It's an extremely powerful tool. Ingenuity Pathway Analysis has been known for years now, 15 years, about this interaction network. It's basically connecting the molecule together in a way that they belong to some kind of biological processes or disease, similar disease. And you can overlay many things here. And finally, I'm just glossing that over. If you have particular findings, we can import this finding yourself in our knowledge base, and only you will be able to see that. It's a very cool thing. So today, for the example, I received that QIAseq RNA panel about breast cancer cell line, and they basically have knocked down particular tumor suppressor, which is the transducer of HER2, top B1, and we are going to understand that transcriptome. So we are going to look at what are the signaling and metabolic pathways that are involved, in what way are they activated, inhibited, and we're going to use the canonical pathways features. We're going to understand what is the underlying transcriptional program using the Epson analysis, and I'm going to show that to you in a couple of slides. We are going to question what are the biological processes are involved, and in what way, and we're going to use the disease and function features. I'm going to just show that, but it is not quite adapted for now to that particular sample today, but we can identify isoforms that are differentially expressed and are connected to cancer using the isoprofiler, and we can draw many new hypotheses using this powerful network that I can explain today. So this is the typical summary we received from Melanie. Right? She had that tab, that Excel spreadsheet basically, and we have an ID that we can map to a gene symbol, that we know what is the gene. We had a p-value because there was replicate in that samples, and we have the fold change between the two experiments. It was with the knockout of top one versus the control. So this is the typical data you have uploaded. It's very straightforward. I don't need to think about complexity here. I just upload it, and we are going to proceed to the core analysis. So when you receive that data set, you push, you set up your cutoff, you set up your p-value, your fold change, your FDR if you have that, and IPA is going to basically process everything in the background and display that to you. Like that, you can pay attention only to the biology and not be worried about anything else. The analysis in IPA are based in many different tabs. It's very simple. We have a summary that explains what are the top canonical pathways, some top upstream regulator, some top causal networks, and many other things I do not show here. We can look at specific canonical pathways, some upstream analysis in the third tab, disease and function, and so on. So it's very simple. These tabs are somewhat independent, but yet interconnected because of the biology. This is a typical example. This is one of the cell line. We have knocked down the top one transcription factor, and the question I ask is basically what are the canonical pathways that overlap with the data you have, right? We are talking about 300 genes in that QIAseq RNA panel, so it's somewhat limited but very powerful. Like that, we can narrow down the exact understanding on the biology, and we would like to determine if any of these pathways has some kind of activity, has been turned on or has been turned off. And this is what we call pathway activity analysis. So in that particular example, you can see that the cell cycle G1S checkpoint regulation has a strong overlap. So many of the genes in that RNA panel overlap with that particular canonical pathways. And we can determine that the activity of that pathway is probably turned down, and this is based on the expression pattern that we can overlay on this particular pathways. Cyclin and cyclin seems to be turned on, p53 signaling seems to be turned on in that particular case, and many other things. So in one glance, you can understand a lot of the biology here. This is the typical cell cycle G1 checkpoint regulation that we think, based on the pattern of expression of the genes that you can see in red and green, we think it's predicted to inhibit it. In other words, that canonical pathway has been turned down. So it's a very key information we understand here. When you knock down the top one transcription regulator in that BT549 cell line, which is the breast cancer cell line, it's a Claudin low type cell line, so it's very invasive, we're able to shut down that checkpoint regulations. And another point that I may add is what you see in gray here is basically genes that are present in your data set but didn't pass the cutoff. So let's have a better look at what's going on in the biology. We know what are the pathways that overlap, and we know what type of activity this pathway. Now the question we'd ask is, who is driving all of that? Based on the pattern of expression of the genes that you see here that are known to be downstream of this particular regulator, we basically are able to answer the question, okay, based on this 5 gene, THPO, which is a cytokine, seems to have turned on a program that led to these expression pattern of these genes. So basically, based on these expression pattern, we consider that THPO, which is a cytokine, has activated a program, and the z-score seems to be positive here. So we can quickly look through all the type of molecule we have, and here I mentioned kinase, microRNA, transcription regulator. I can see, for instance, that the TP53 here, which is a tumor suppressor, which is present in your data set, and you can see that with that upregulation, when we are able to determine, this is the transcriptional level, we are able to determine the functional activity. And based on the pattern of expression of 33 genes here that are known to be downstream to TP53, based on the pattern of expression, we're able to say that TP53 program has been turned on. So you have the transcriptional aspect here, and you have the functional aspect. It's extremely powerful, right? And this is a simple network where you show the prediction of TP53, known to be activated in that case, and in the outer ring here, you see the real genes with their expression that you have obtained in your QIAseq RNA panel. So CRT1 is downregulated in your sample versus your control. TP73 is upregulated. And we're able also to overlay something really powerful is, does that prediction that we think, based on pattern of expression of the gene, the prediction of activity of that particular transcription factor, does it fit the literature? Does it fit what we know in the literature? And we basically color code. Here we say it fits the prediction of activation when it's orange, it's an activation. So TP53 is known to activate DDIT3, while DDIT3 is upregulated, so therefore there is a strong prediction of activations. When it doesn't fit the reality, we show it doesn't. It's finding inconsistent with the state of downstream molecule. When we can't predict because the writer or the authors of paper didn't precise exactly the direction, we say to the biologists that we can't predict that effect. So it's very clear now. We know TP53 is predicted to be activated. We know the pattern of expression of the genes known to be downstream of that. And the question I will ask now is, okay, what are the disease and function? Basically, what are the biological processes that seems to be relevant to that particular network here? And we understand, as a biologist, this is a couple of key hypotheses that you can test and validate in the lab. And here, basically, based on that pattern of expression, right, we said TP53 program is turned on. Well, it drives the apoptosis up, which is represented by an orange graph here, and the proliferation of the cells are down, right? So if TP53 is working well, known as the tumor suppressors, you will drive the apoptosis, and you will limit the proliferation of cells. These are very key phenomenon because you can test all of that in the lab, right? So this is a new hypothesis. Let's complicate a little bit of that situations. We all know that regulators that you see never works alone. We talk about TP53. I selected another one, beta-catenin 1. We try to understand now that a network of regulators together. So the one that you see here, all of these guys are regulators, right? They are connected to each other by many types of relationship, including activation, inhibition, membership, RNA binding, DNA binding. All of these guys together are working actually together and may explain the pattern of expression of 38 targets downstream. They are not shown here. I didn't show that. But the bottom line I'm trying to explain here is the fact that we know that regulators work together in connection and they are intertwined to make the expression of genes downstream realistic. And this is another type of hypothesis that you can test in the lab. Catenin beta 1 activate P38 map that activates SMAD3, and that SMAD3 may be shown to maybe targeted, maybe connected to, maybe connected to many target downstream. This is an actionable hypothesis. You can test that relationship. So right there, we are decomposing the biology, trying to understand what are the relations between all these regulators together and their relative target downstream. Another type of level is to look uniquely at what we call the biological processes. This is called a downstream effect analysis. But basically, based on the pattern of expression of the gene that you see here, we're able to determine that, yes, the apoptosis of tumor cell line is increased. That's a key phenomenon. If you are back in the lab, you would like to understand, oh, I'm studying apoptosis and I am studying VGFA and XBP1. What are the relations with that particular phenomenon? Well, this is an hypothesis that you can test. Here, this is a prediction. Here, this is the real pattern of expression of gene. I insist on that. This is a prediction brought by the fact that all these genes are known to be downstream and associated to apoptosis. And based on their pattern of expression, we establish apoptosis is increased. Another level of complication, and I think it's probably the most powerful network, is what we call a causal network. So a causal network is something that we came up 2 years or 3 years ago now. And by the way, we have a strong publication that is highly cited. And what it is the relation between network, between regulator together in a very causal way. So we draw that network by establishing only activation and inhibition edges between these regulators. So it's very causal. There is no sidetrack. We know that one activates the other one and the other one inhibits. Based on that pattern of expression, right, we're able also to tie that to a set of genes downstream. And this is in fact based on the pattern of these genes, CDK1, AKT3, which is downregulated in your sample. We're able to establish that pattern of activity of these particular regulators together. So what it says here is TCB3 is a master regulator of the causal network. And it works in association with 5 others, 6 others regulators to drive the expression pattern of many genes downstream. Just a couple of them are shown here. So that's the first part. Everything is causal. Regulators together are connected by activation inhibitions, and the set of regulators here are connected to the set of the target downstream by only expression and inhibition. So it's very down to the wire, right? We are not basically kidding about the hypothesis we like to bring. And the second point is, I asked the question, okay, you said to me that the master regulator TCB3 is extremely important to explain the vast majority of expression pattern of gene downstream, but I would like to understand what is the relation with invasion of cells, right? We are understanding the role of these breast cancer cell line invasions. Well, show me what are the intermediate between invasion of cells, which is the disease and function I would like to score against. It's called scoring against. Show me the intermediate that can connect this TCB3 to invasion of cell. And the example here is that cyclin inhibitor, which is upregulated in your samples. So you understand here, this is a complete new hypothesis. Maybe TCB3 has never been connected to any of the known targets, so we're filling up with regulators. We drive the system by only activation and inhibitions. All of that sits on expression and transcription edges. So it's very, very causal. And now we are tying that to biological processes. That is the key hypothesis that I would like to test in the lab. Now let's look at another phenomenon that we can, that I mentioned in my first slide, is connecting everything together, which we call regulator effects. So it's a very simple drawing. You have your target downstream coming from the QIAseq RNA panel, all up and down. We were able to determine what upstream regulators and what their level of prediction. Catenin beta 1 is upregulated. This one is downregulated. And we are able to tie that to the disease and functions. So you understand that is another set of hypotheses that you can validate and test in the lab. I mentioned at the beginning that the QIAseq RNA panel that Melanie has given me look at the gene level. But let's say if we wanted to look at the splicing variant or isoform level, that isoprofiler allows us to discover isoform that may drive the tumor progression in that particular cancer. I selected, just for the example, another type of cancer. I could have selected a breast cancer disease, and I will be able to ask the question, okay, show me in my data set what are the splicing variant and their regulation, their relation with the particular disease. So this is a tool I didn't use particularly for that QIAseq RNA panel, but let's see in the future if we can use that. So just to resume the situations, not being a bioinformatician, but using QIAGEN bioinformatic approach, I've been able to look at the differential expression in 2 breast cancer cell lines in a knockdown experiment of top 1. We were able to understand the signaling pathways and what way they are involved in the tumor progression cell line. We were able to discover the potential program that can be either induced or repressed in this breast cancer cell line. We were able also to discover probably new biological processes that participate in that tumor progression. In that particular case, I show the identification of new isoforms that are differentially expressed and they're connected to a particular cancer. I select another one here. And mainly, and I think this is the most important, beside the description of what you have in your data set, is bringing and highlighting new hypotheses that you have to test and validate in your lab. And I select one here and trying to understand what are the new hypotheses that I can draw that can explain the cell invasion. So it's extremely powerful tool in terms of description, confirmation of what you have in your data set, that's your expertise, but also highlighting new hypotheses that you will never be able to draw on if you were not using that particular software. So to that, I think I'm going to thank a lot Melanie Usang for that beautiful presentation, and I think I can hand that to Krishnan to finish that webinar. I hope you have enjoyed that presentation.
Thank you very much, both Melanie and Jean-Noel, for this fantastic presentation. It is just fabulous that how we have put together this whole story of starting from sample to insight, where Melanie went through some of the how do you move from samples to data analysis, and Jean, how you took the data analysis and how it goes from how you can make a sense out of the complex data that we normally see in next generation sequencing. Of course, there are many questions. First, I want to inform the audience that feel free to submit your questions now so that we can at least add it to the queue. If not, we'll try to answer as many questions as we can, but in case we are unable to answer, we'll send you an email with all the answers. And this will be, we'll forward these questions to the speakers. With that, let me ask you the first question. The first question is for Melanie. How many pathway-focused panels currently do we have?
Well, at the moment we have 8 wet batch lab tested panels, but you can, there's 170 plus pathway and disease focused panels from the R2T squared, which consist of 84 gene panels. You can make your own pathways and customize your panel. So, but there are 8 that have been wet lab tested at this point.
Okay. All right. With that, the second question for you is, again, this is also for Melanie. Can we get this gene list for these panels?
The gene list should be available soon on the QIAGEN.com website, but we could also email you the gene list in the meantime.
Okay. Okay. Thank you. The third question for you would be for, are these all for plants? Are these available for plants also?
At the moment, it's only human-based, but if you're interested in mice, rats, any other organism, I'm sure we could try and get primers designed and make it happen for the organs that you're interested in.
Okay, perfect. The next question is for Jean. Are there any R packages for digital RNA-seq?
The R package?
R, it's called any R package for digital RNA-seq.
I couldn't understand the question, but the point is, regardless of what we receive from the QIAseq RNA panel, that gene list that we have on an Excel spreadsheet, we're able to upload it. So we are not limited per se on that level on our side. I hope this is what the people are asking.
Okay. The next question, thank you. The next question for you is there a difference between the causal network and downstream network?
Yes, absolutely. So what we call a downstream effect analysis, which will be seen under the tab disease and functions, look at the biological processes and their prediction of happening, or they increase or decrease, and this is based on the pattern of expression. That's one point. Now the causal network that I show is basically trying to understand how some of the regulators that are only connected to each other by activation inhibitions may plausibly drive the expression pattern of target downstream that are present in your RNA-seq panel. And that causal network, we can ask the question, is it driving, increasing or decreasing a particular biological processes of matter? So it's two different views of the biology. One is understanding the biological processes, and the other one is bringing a new hypothesis of how regulators can drive a particular biological processes.
Okay, thank you. The next question for Melanie is, did you mention from cDNA synthesis that you can go directly to library quantification? This is all in a single kit.
So yes, it's from cDNA synthesis up until the final purification step just prior to the library quantification. There is nothing in the kit for library quantification, but in the QIAseq targeted RNA panels, you do get from cDNA synthesis all the way up until the final purification step.
Okay, thank you, Melanie. The next question is for Jean. IPA could be used as a CLC plugin, free of charge. Is it compatible with plants, or can we? Are there any other species that can be used?
Okay, so there is many questions in there.
Yes, I know.
First of all, just one step back. What is that CLC? CLC Bio is basically has developed many very interesting software that do the data analysis, and this is now part of QIAGEN for the last 3 years. And yes, we do have a very smooth interaction also with that biomedical Genomic Workbench, this is the name of the software, that allows the processing of RNA-seq data set and push that data to IPA. So we have, it's probably the topic of another type of webinar series, but this is a very strong connection that we have right now with that software too. You have the data analysis being processed in Biomedical Genomic Workbench. You process from the FASTQ file to the differentially expressed level of each of the genes at the gene or the transcript level. And using a plugin, we push the data the same way we have done with Melanie into IPA. We upload the data set and we start the biological interpretations.
Okay.
So everything I have to mention, everything is on the QIAGEN bioinformatics.com website, and you can look at the extent of all our software and bioinformatic tools we have right now, and a couple of explanations of how they are and what do they do.
Okay. All right, that was the last of the questions for today. And so I want to thank both the speakers for today for their fantastic presentation, and all the audience for joining us today. I know there are some questions that we have not answered today, but what we'll do, since it's we are past the time, I want to stop the webinar here, but we'll send you answers to your questions as soon as possible, within 48 hours. With that, thank you very much, and you all have a wonderful day.